Hello and welcome to today's WTG webinar session. Our topic today is precipitate and solubility investigation, a case study for an active pharmaceutical ingredient using high throughput experimentation. And our speaker is Matthew Bahr. So let's start with an introduction to Freeslate. Freeslate delivers products and related services for high throughput research. For more than 20 years, they have automated research in the pharmaceutical industry. With over 100 systems in the US, Europe, and Asia, Freeslate's laboratory automation platforms and powerful software enable dramatic gains in R&D productivity and innovation. To learn more about Freeslate solutions, please visit www.freeslate.com.
So let's begin by talking about the specific drug that we investigated, which is the primary focus of this case study. So as you can imagine, I can't reveal any proprietary information about this particular drug, but what I've done is I've given it a nomenclature of GSK 123. It's completely made up, but the data that you're going to see is taken directly from the experiments that we ran using the Freeslate robots and the support equipment that goes in with our automation lab. So several years ago, GSK advanced an asset into clinical trials. And for the second biostudy, the tablet formulation was manufactured using a traditional wet granulation process. The tablet was composed of the salt form of the API with a 31.5 milligram API dose into a 200 milligram tablet. The first biostudy used a similar tablet formulation and API dose, but the original tablet weight was 800 milligrams. In addition to a change in the core tablet weight, several batches of tablets were also manufactured using different excipients. Namely, the binding agent and the disintegrants were replaced with different materials. So, for the different batches manufactured for the upcoming clinical trial, some analytical testing was performed. And in this slide, what we're looking at is the dissolution data for the original 800 milligram tablets, and then also the new 200 milligram tablets. For each of these two tablet sizes, sub-batches were manufactured with and without sodium lauryl sulfate, which is an additive in the tablet formulation. As you can see, the 800 milligram tablets perform better than the 200 milligram tablets did, but the presence of SLS had no real apparent effect on the dissolution profile. This slide is similar to the previous slide, but in this case, what we're able to do is compare the impact that the two different binders have on the dissolution rate. What's interesting to see in this slide is the difference in the dissolution rate that HPMC has when compared to PVP. When HPMC is used in the tablet formulation, the rate is faster, and the overall dissolution is more complete than when PVP is used in the formulation. So the difference in bioavailability between the 200 milligram and the 800 milligram tablets can possibly be attributed to a combination of several factors. At the time, it appeared possible that the salt form was converting to the parent free acid. If this was the case, then we needed to investigate how various excipients could influence not only the rate of free acid formation, but also the extent to which it happens. It was important to determine if we were seeing the results of precipitation inhibition. So the most influential excipient in the drug bioavailability appeared to be HPMC, but we couldn't really rule out the possibility of other ingredients also contributing to the dissolution profile of the 200 milligram tablets. There we go. So now that we've introduced the background to the API, on the API that is the focus of this case study, we'll take a deeper look into how high throughput experimentation was used to help gather data in an effort to learn more about the solubility and the API's propensity to precipitate out. There's a lot of information on this slide, so let's go through it. A Freeslate CM3 was used to perform the studies that are described in the rest of this presentation. At GSK, we purchased a core module 3, also known as a CM3, in 2014 for our King of Prussia site in Pennsylvania. In the time since, we now have a collection of 3 CM3 machines as well as a protege unit for solid dispensing. 2 of our CM3 robots are outfitted primarily as pre-formulation machines, and they perform liquid and solid dispensing, and they have the capability to measure pH, and one of them can also process viscous liquids. Our third machine has an optimization sampling reactor module, or an OSR. This is dedicated to chemistry reaction screening. Our lab is managed by Ken Wells, and we have 4 full-time scientists as well as a Drexel University co-op student contributing to the workflow. In addition to that, we have scientists from around the organization who contribute to the various work packages on a part-time basis as needed. So for the first set of experiments that we're going to look at, the focus was on measuring the solubility of the API in 5 different liquid media, as you can see on the left side of your screen. 2 were simulated biomedia, and the other 3 were more traditional buffer solutions. In each of the media, the goal was to dispense the API along with 6 different formulation excipients into a vial and then stir under specific conditions and then ultimately measure solubility. Before we could start the work package, we had to address several complicating factors. The API has a very low solubility. There was already evidence to show that the salt form of the API changes to its parent-free acid. And lastly, the API is a chelating agent, and any experiments that we conducted would have to be done with no metal components in direct contact to the API. So as you can see in the photographs below, the chelating effect was not evident until after we performed our first set of experiments using standard stainless steel stir bars. In the picture on the left side, you can see how the iron reacted with the API and formed a rust-colored complex. We eliminated this by purchasing Teflon-coated stir bars, and you can see on the photograph on the right how the API on the bottom of the vial remained pure white after stirring for several hours. Here's a brief explanation of what the library consisted of. And this photograph shows a traditional 96 well plate that contains 1 ml vials. This is typical to what we use in our lab. Into each of the 1 ml vials, we added 5 milligrams of drug. The goal was to dispense more API into the vial so that we could use the solids that remained to perform solids characterization tests. In addition to the API, we then dispensed various amounts of formulation excipients into each vial, ranging from 0 to 7 milligrams of excipient. And into each vial, we then added 500 microliters of biorelevant media, or the pH buffers. Each test condition was performed with 5 replicates. The tests were run for 3 hours, and we ran them at 37 degrees centigrade. This time and the temperature were selected to closely resemble the stomach retention time of an ingested tablet at body temperature. The 96 well plate was centrifuged to force phase separation between the saturated solution and the excess solids that remain in the bottom of the vials. And then following a filtering process, the solutions were placed on an Agilent 1100 series HPLC, and we measured them for solubility. The solids that remained in the vials were then dried and sent out for Raman analysis. The experimental plan that you see here has 1,200 data points associated with it. So you can see that this is a very detailed and complex work package that requires a considerable amount of data manipulation. So at this point, we're going to stop briefly for our first poll question, and you'll have 1 minute to answer, to provide an answer on the right side of your screen. When we conduct lab automation experiments using high throughput experimentation, we have the opportunity to perform our experiments with replicates. And those replicates provide statistical significance to our data, and it also gives us the opportunity to determine if we have any outliers in our traditional data points. So when we perform manual experiments at a very early phase, we often limit ourselves to a single sample point, because sometimes manual experiments can be very time-consuming. So when considering high-throughput experimentation, what do you think would be the appropriate number of replicates to have during experimentation? And okay, so we'll move on to the next portion of this presentation. Okay, so now that I've explained what our plate design looked like from the previous slide before the poll question, let's take a look at an example of what those 1,200 data points can provide us. In this slide, what we're looking at is the drug concentration in simulated gastric fluid when 6 different formulation excipient powders are added to each vial. Clearly, the drug is not very soluble in SGF. Even as we add large portions of excipient, the solubility remains largely unaffected. However, there is clearly one trend that is different from the other 5, and it's HPMC. HPMC is a binding agent, and it seems to increase the drug concentration in solution significantly as a portion of the HPMC increases. And you can see the contribution of the excipient on the x-axis. There's 240 data points on this particular slide, and they provide very good insight to our drug's behavior. The same plots were also prepared for the other 4 media that we tested. And I'm highlighting SGF primarily because it's the most relevant media to represent the conditions at the drug's administration site. With this data in hand, we proceeded to perform additional investigations in order to hopefully identify what exactly is happening here. In each vial that was prepared, there was a certain portion of undissolved drug that remained in the bottom of the vial. These solids were removed from the biorelevant media and dried. The dried material was sent to our analytical development lab, where XRPD was performed on each sample. The patterns that follow give evidence that there is form change actually taking place. In order to prove that there is form change, we first started with a sample of the free acid drug form. The free acid was subjected to 37 degrees centigrade for 3 hours in the 5 different media. And as you can see, there is a clear peak in all 5 solutions, which shows us the baseline at which point the free acid is present. Now in this slide, we see something very different. This is the XRPD pattern for the salt form of the drug at 37 degrees C after 3 hours of mixing in the same 5 media as in the previous slide. On the right side of the XRPD pattern, the salt form is expressed. It's important to note that the amplitude of the pattern is not the same for all 5 media. On the left side of the pattern, at around 5.5 degrees, the presence of free acid is evident, even though no free acid was placed in the vials at the start of this particular experiment. What this tells us is that after 3 hours, some of the salt form converts to the parent free acid. And in the case of SGF, which is the red colored trace on the screen, the conversion from salt to free acid is complete. For the next portion of this webinar, we'll look more closely at what has been done to better understand what might be behind the form change for this particular compound. For our form analysis investigation, we needed to discover how many forms this drug has in biorelevant media. So a series of experiments were designed. We have 2 experiments using 2 different known forms of the salt. We dispensed the drug into 1 ml vials along with the 500 microliters of SGF. The vials were stirred for 1 hour for the first set of experiments and 4 hours for the second set of experiments. And then all the experiments were conducted at 37 degrees C to mimic body temperature. In order to have a baseline for the form analysis, an identical set of experiments was also conducted using the free acid drug form. All of the sample wells were centrifuged and then filtered. They were measured for post-test pH and then placed on an HPLC for solubility measurements. The residual solids that remained in each vial were prepared for form analysis, but this time using a thermoscientific FT-Raman spectrometer. And this particular spectrometer is designed to work with 96 well plates. So here we see the results of the solubility and pH measurements. The plots on the left represent the 1-hour data, while the plots on the right side of the screen represent the 4-hour data. So on the left side, we see orange, dark gray, and light gray bars, and these represent the free acid solubility, while the blue, the red, or purple, and the green bars represent the solubility of the salt form. So from this plot, we can see that the salt forms 3 and 4 have higher solubility at 1 hour than the free acid forms 1 and 2. However, after 4 hours of mixing, on the right side, the solubility of all 3 batches of the salt have dropped and are essentially equivalent to the solubility data generated for the free acid. The cause for this is likely due to the fact that after 1 hour, the salt form has not completely changed to free acid, but after 4 hours, the salt has converted fully to free acid and precipitated out of solution. So we'll look at the FT-Raman spectrograms on the following slides to back up that conclusion. But before we do that, we have another poll question. So we have another 60 seconds to answer this question. There are many ways that we can analyze data. And the most basic and probably the most common way to look at data and generate visual representations is to use a spreadsheet. However, I've been seeing more often that folks are using statisticians or statistical software as an aid. One example is Spotfire, but I know there are many others, and I know that some folks simply report the raw data and perform no statistical analysis at all. So I'm interested to know what you guys are doing with your data when you generate it. And we'll take about 20, 25 seconds to answer that question. So as I mentioned, we're going to look at several FT-Raman spectrograms. I have 6 of them to show you, 6 slides of them. So in this first set, we're looking at the reference standards for 3 different batches of the salt. One is known as having form 3, while the other 2 batches were manufactured as form 4. The reason for the 2 salt forms is due to the fact that the crystallization process is slightly different between the 2, causing a slight form change, although both of them are actually the same salt. It's important to point out that there are some limitations to the FT-Raman sample preparation at this point. Although I'm able to stop the mixing process and reduce the temperature, it still takes some time to isolate the solids from the liquids in the vials. And so what the data you're actually seeing is sometime beyond the 1 hour time point. I call it 1 hour data, but it's a little bit past that for the amount of time it takes to prep the sample. So we hope to eliminate this issue with a new 96 well plate XRPD, which we've recently purchased, and it's intended to analyze samples nearly immediately after the solubility samples are extracted. In this set of spectrograms, we're looking at salt forms 3 and 4 after mixing in SGF at 37 degrees for 1 hour, or just past 1 hour. Both batches of form 4 are identical to each other, but there are some definite differences from the form 3 batch. I overlaid the 1 hour form 4 data from the previous slide with the form 1 reference spectrogram. Here we can see that both batches of form 4 have converted to the free acid form 1 after about 1 hour, or just past. This data corroborates with the solubility data shown a few slides ago. Now we'll look at the 4 hour data and see what conclusions we can draw. This set of spectrograms show the form 3 and the form 4 scans in comparison to the form 1 scan, which is the reference standard at the bottom. It's similar to the previous slide, but this is the 4 hour time point, and all 4 scans look practically identical. In this particular plot, I overlaid the 4 hour data of all 3 batches from the previous slide with the form 1 reference spectrogram. The light blue scan is the reference standard for form 1. And with this, we can see that all 3 batches have conclusively converted to the free acid form 1 after 4 hours. Up to this point, I've pretty much deliberately left form 3 data out of the main discussion from the previous spectrograms. This is because there is something rather unique happening with the form 3 that requires further analysis and discussion. So with this set of scans, in the top scan, we're looking at form 3 after 1 hour. And the 3 following scans are the reference standards for form 3, form 2, and then form 1 at the bottom. By visual comparison, it's clear that we have a form change that has occurred after 1 hour. The form 3 scan most closely resembles the form 2 free acid standard reference standard. So we know that we have a conversion from salt to free acid, but not to the most stable form 1. So what is happening after 4 hours? And let's take a look at the next slide and see. Similar to the previous slide, we have a spectrogram of form 3, but after 4 hours of mixing. The other 3 scans are the same, 3 reference standards from the previous slide. And in this slide, it's clear that the full conversion to form 1 has taken place. From the FT-Raman data, we can conclude that, first of all, FT-Raman is a good technique that can be used to determine drug form. In the experiments performed for this work package, the solubility data indicated that solubility of the salt dropped after several hours of mixing, and this could indicate that some of the drug was precipitating out of solution. By applying FT-Raman, we could see that form 3 converted to form 2 free acid after 1 hour, and form 1 free acid after 4 hours. And we were also able to determine that both batches of form 4 converted to form 1 free acid after only 1 hour, and remained as form 1 even at the 4 hour mark. From the solubility data and the spectrograms, you saw additional data for the free acid that I didn't really discuss in any detail. However, with the use of high throughput experimentation, we were able to run additional experiments that were similar to the ones I just discussed, but this time using free acid. And in addition to that, we enhanced the free acid experiments to include the addition of only 1% HPMC to see to see what effect HPMC had on the solubility of the free acid. By using the Agilent 1100 HPLC and the thermoscientific FT-Raman, we concluded that Form 1 is the most stable form of this API. With and without HPMC, the drug remains as Form 1. However, the Form 2 batch converted to Form 1 after only 1 hour of mixing. And when we added 1% HPMC, which is such a trace amount, really, we found that the Form 2 API did not undergo a conversion even after 4 hours of mixing at 37 degrees. So for this last section, I'll talk about some experiments that were performed to support mechanistic understanding. And with that, what we'll do is pause for our third and last poll question. And we have 1 minute to answer this question. Do you use a DOE package in combination with your HTE experiments? And really, I'm just looking for a yes or a no. We've seen in previous webinars from Freeslate, I think Justin gave one earlier this year, that you can use DOE software to help run complex experiments that have multiple variables and responses. So I'm looking to find out if you guys use DOE software packages to help with your designs, or do you tend to do you tend to simply write your experiments up in Design Expert, kind of the old-fashioned way? So we'll take about 15 seconds to answer this question, and then we'll move on to the final part of the presentation. Okay, here we go. For those that responded to the poll questions, I really appreciate it. Okay, so our last set of experiments looked at equilibrium solubility of the salt and free acid in the presence of varying amounts of HPMC. By looking at this, you can see the relevance of the poll question that you just answered, because some of these experiments can be pretty complex. We dispensed a known amount of the salt or the free acid into 1 ml vials. For the media, we added 500 microliters of SGF, and we spiked that SGF with increasing amounts of dissolved HPMC. The experiments were spanned from 1 hour to 24 hours for the free acid experiments, and then actually we went all the way out to 168 hours for the salt form of the drug. This data shows the solubility and pH of the free acid. What's most notable in this data is that at the 5 milligram per mil HPMC SGF solution, the equilibrium solubility is extended to at least 24 hours and possibly beyond. The 10 mg per mil solution supports this as well. But at the lower HPMC concentrations, the equilibrium solubility is established within about 4 hours or so. HPMC clearly has an effect on the equilibrium solubility of the free acid, and the presence of HPMC increases the drug concentration in solution. This data is equally interesting, though there's a lot more data on the screen, so I apologize for that. Here what we're seeing is that the equilibrium solubility is established in about 1 hour, and that's just in the presence of SGF alone, with no HPMC. In small amounts of HPMC concentration, up to about 0.2 mg per mil, the solubility decreases over time and then reaches equilibrium solubility sometime between 24 hours and 3 days. This is expected because we know already that the salt form of the API is precipitating out as free acid, which has a lower solubility than the salt form. However, when significant amounts of HPMC are added, such as 5 mg per mil, the precipitation appears to be inhibited by the presence of HPMC, and the equilibrium solubility is extended to at least 24 hours and even beyond. Some of the 72-hour and 168-hour data points could not be collected, unfortunately, due to the fact that the sample vials dried up and we just didn't have any material to test. And that's really because of the elevated temperatures that we were running these experiments at. Here's a graphical representation to support this analysis. At 1 and 4 hours, the salt has a relatively low solubility. This indicates that these short time periods, the salt remains stable. However, at the 24-hour time point, the solubility rises much higher. This graphically demonstrates that the salt remains stable and does not precipitate out for at least 24 hours at the high HPMC concentrations. This graphic further supports that increasing the amount of HPMC inhibits the precipitation of the salt into free acid. If the salt did precipitate out at 4 hours, then we would see lower values of the drug concentration in solution. So essentially what we're doing here is just looking at the data in slightly different ways and trying to interpret what those results are. This slide shows the 24-hour data of both the free acid and the salt. It shows that increasing amounts of dissolved HPMC in SGF increases the concentration of the drug in solution. But at 24 hours, the presence of HPMC cannot continue to inhibit the precipitation of the salt form any longer. This provides tremendous insight into the mechanistic behavior of the drug in simulated gastric fluid. So with that, now that we've discussed the final set of experiments, let's take some time to summarize the entire body of work that I presented here today. We've seen that using SGF, simulated gastric fluid, as our primary media at 37 degrees centigrade to measure solubility of this drug is relevant to mimicking human body conditions, and it closely represents real-life conditions. So we should continue to use this as a method in the future. The data indicates that excipients can indeed impact the solubility of an API, and that HPMC, in this case, significantly increases the drug concentration solubility of this particular drug in simulated gastric fluid. It appears that the drug needs to dissolve in close proximity to the excipients in order to have a high likelihood of forming a complex with the HPMC in this case. This new species can help the drug stay in solution and can inhibit the formation of free acid. We've demonstrated that by using HTE in combination with FT-Raman, that we are able to rapidly track the progression of polymorphs with a small amount of solids material. Yet at the same time, we can produce a large volume of data. FT-Raman and HPLC can be used together to verify form change. The data generated from HPLC allowed us to target experiments and then pinpoint where the form change is occurring. With this knowledge, we could use the FT-Raman spectroscopy to support the HPLC data conclusions. In this particular case study, we analyzed several tablet formulation excipients and the impact that they have on drug solubility. We found that HPMC significantly affects the drug behavior in that it extends the equilibrium solubility of both the free acid and the salt. And in addition to extending equilibrium of the salt form of the drug, we found that very large amounts of HPMC also can significantly prolong the formation of the free acid precipitate. So with that, I'm going to close my presentation, but before I do, I'd like to acknowledge several GSK scientists who contributed to the data that I shared with you today. From our automation team in Upper Merion, I'd like to acknowledge Ken and Chris. And we also have amazing contributions from our Drexel University co-ops. They have a great program, and we have a real benefit of having a co-op student every 6 months come through the rotation. So I'd like to thank Dan and Sophia, our previous co-ops, for providing a lot of support in the creation of these experiments. This work was done in support of our product development partners who are focused on developing a robust tablet formulation. So I wanted to thank Shivari, Esteban, and Shanming for their contributions to this particular project. There are a bunch of others who contributed to progressing this asset through formulation development and onto clinical trials. Yet unfortunately, I can't name them all, but the dozens of highly skilled scientists at GSK hopefully know who they are. With that said, we have some time for a few questions. I want to thank everybody for attending, and if there are any follow-up questions that I was unable to answer on the line here today, feel free to contact me directly. I have my email address at the bottom of the screen. And at this point, Louise, I think I could turn it back over to you.
Fantastic. Thank you, Matthew. So let's wrap things up with our question and answer session. If you have any questions for Matthew, please submit them into the question and answer dialog box at the bottom right of your screen. We've already received a number of questions, which is great. So, we're going to do our best to get through as many as we can. So let's start with the first question. So, Matthew, on slide 9, you mentioned that the drug chelates, and you showed a picture of the rust color in the vials. Did the solubility data for these look different than for experiments that you've ran with the Telfon coated stir bars?
Yeah. So So what this question's asking basically is, we started our experiments with standard stainless steel stir bars, and then we found that that the drug was chelating and everything turned in this dark red color, and then we stopped those experiments and moved on to using Teflon coated stir bars for the remainder of our experiments, and this stopped the rust formation. So when I looked at the solubility data for the experiments that we ran that chelated, we found that the solubility values were still about the same as they were with the Teflon stir bars, but on the front end of the HPLC data, we saw we would see spikes that would indicate, it seemed to indicate that there was some sort of a complex being formed or a degradant present in the solubility trace. So the data still looked fairly valid, but we elected not to use any of that and basically started from scratch, moving with the the Teflon coated stir bars.
Okay, interesting. So I've got another question here actually. So on slide 32, you show n equals 4 and n equals 8. Do these represent the number of replicates that you ran? If so, why did you run only 4 replicates at the 1 hour time point?
That's a good question. So what we're looking at here is we have on our Freeslate robot, we have 4-tip needles, and we also have 4-tip pH probes. And what this means is we can do dispensing with 4 tips at a time. We can do filtering with 4 tips at a time, I should say. And we can also measure pH at 4 tips at a time. So I tend to run experiments in replicates of 4 or multiples of 4. In the case of the n equals 4 on the first experiments, it's mainly because of the amount of time that it takes to collect that the samples. And then with the later experiments that take, for example, 4 hours or beyond, I have a little bit of more flexibility with the amount of time because it's a 4-hour experiment. So I have a little bit more wiggle room with collecting the actual samples.
Okay. So we've got another question here. It's quite a good one. On slide 12, you showed data with the various excipients being added up to 7 milligrams per vial. Why is it that you stopped there instead of, continuing to higher amounts?
So that really, it has to take into consideration a few things. First of all, we ran I think I mentioned 1,200 data points in that set of experiments. That's quite a few plates that we had to put together in order to collect that data, and that process took us several weeks or more actually of full time on 2 CM3 machines. You have to consider the amount of excipients in the tablet formulation itself. So if I have a 31.5 milligram dose of API in a 200 milligram tablet, I want to sort of ratio the amount of HPMC that I'm including in my experiments on the Freeslate machine so that they are in context with what that formulation looks like. So essentially, if I went beyond 7 milligrams in each of those vials, it really wouldn't be a realistic representation. I need to consider what would be a realistic representation of the drug to excipient ratio that's in the tablet formulation.
Okay, excellent. Just one more question here for you. On the 18th slide, your data begins at 1 hour. Have you tried to shorten this to 15 or 30 minutes at all?
This question actually relates pretty well to one of the previous questions. When I mentioned n equals 4 or n equals 8, that's the number of replicates that I'm running for each of those experiments. The reason why I pick n equals 4 for a 1 hour experiment is because it does take some time for the robot arms to move over to the source plates and then to withdraw the liquids, bring them over to the filter plate, run that filtering process, and then go on to the next set of sample wells. if that process takes too long, if that process of filtering and daughtering and diluting takes more than 10 or 15 minutes, then I'm really no longer looking at a 1-hour experiment. I might be looking at something that could be 1 hour and 15 minutes. So I need to really keep that data as close to the time point target that I can. If I try to run these at 30 minutes, then I'm really looking at a large window of time that it would take to run those replicates at very short time frames. So dissolution testing is probably very appropriate if you're looking to see the drug concentration profile at very short periods. But we're really not looking at using this machine as a dissolution apparatus as much as just measuring the solubility. So while we can take short time periods, we have to really keep that realistic, and we really probably would only run those with n equals 1 or n equals 2 if we wanted to do it the right way. And then we don't really have statistical significance in our data with so few number of data points. So yeah, really just have to weigh how much time you have to run the experiment, and then how much error you can have in your time point data collection points.
Okay, fantastic. A very thorough answer there. Unfortunately, we don't have any more time for any more questions for this session, but if you do have questions that you'd like to have put to Matthew, please do send them to us, and we will be sure to forward them. Thank you for joining us today for our WTG webinar, and a big thank you to Freeslate and, of course, our speaker, Matthew. Thank you very, very much for taking the time to give a very interesting presentation indeed.